Hello, everyone. Welcome to the Thursday edition of the Red Risk School as we end out our sessions for 2020. My name again is Jeff Willey, Associate Professor of Radiation Oncology at Wake Forest School of Medicine and Director of the Red Risk School. So we are continuing our discussion of the high priority risks to human health and space as defined by the Human Research Program. And specifically this week, we've been focusing on projects that are supported by the Translational Research Institute for Space Health, or TRISH, who partners with the Human Research Program to address these challenges in specific ways, looking for novel, cutting edge, even risky approaches to help mitigate these risks. Now, for several years, neuroocular syndrome has been identified as a primary risk to human health during spaceflight, with over 30% or so of astronauts flying long durations ISS missions presenting one or more ocular disturbances. Mitigating this risk is a priority. And today we'll hear from one investigator who is tackling this concern. Before we get into the introductions, again, some housekeeping. All of the talks are recorded. So if you want to hear this session or previous sessions, please go to Trish's website. These ones will probably be on within a week or so, and you can catch yourself up or see what you missed. If you have any questions, please use the Q&A feature. We'll be monitoring that throughout the presentation, and at the end of the discussion, we'll present some of those questions. We have a little bit more than an hour and 15 minutes total. We'll ask about 15-20 minutes of questions, and hopefully we can get to everything. So with that, I'm going to turn this over to Jimmy Wu from TRISH to introduce today's speaker.
Hello, everyone. My name is Jimmy Wu. I'm TRISH's Senior Biomedical Engineer. And I wanted to talk a little bit about why this risk is of importance to TRISH and NASA. So SANS is the spaceflight-associated neuroocular syndrome that was discovered over the last 10 years from what we've seen with long duration microgravity exposure to our astronauts. And it was so because of that, the leading hypothesis seems to be that because of the zero gravity effects of being in spaceflight, there is a headward fluid shift, and that increased fluid increases the pressure within the skull that might affect the eye. And so this leads to symptoms such as optic disc swelling and other symptoms that affect the vision of a crew member. So the challenge is because this phenomenon seems to only occur in spaceflight, we don't have good models on Earth to be able to simulate those various pressures that human eyeballs may experience in spaceflight. And this is of concern because right now our ISS missions are 6 months. A transit to Mars from Earth would be also 6 months of microgravity exposure. So we would want to understand this phenomenon as we plan and hope to send our astronauts onward to Mars. So with that, we have Dr. Tasneem Sharma, who has been a TRISH investigator over the last year or so. And I'd like to read a biography of her so you get an idea of the fantastic work that she and her on her resume. So Dr. Sharma is an assistant professor at the Indiana University School of Medicine. Her passion lies in the scientific exploration of pathogenic translaminar pressure-induced neurodegeneration, neuronal cell death, and generation of pluripotent stem cells for disease modeling. She utilizes human stem cells to develop retinal neurons and has patented the translaminar autonomous system for studying various diseases to study various her research efforts are committed to driving interventions that can improve public health care. She aims to identify therapeutic interventions geared towards retinal ganglion cell survival through viral gene therapies, CRISPR-Cas9 genome editing tools, cell and molecular biological techniques, bioengineering paradigms, and neurotropic factors. She has published numerous impactful manuscripts, such as a recent one titled Cas9-based treatment of myocilin-associated glaucoma, co-authored by her and has awarded the 2018 Louis Rudin Glaucoma Prize in Glaucoma from the New York Academy of Medicine. In addition, she has received multiple accolades in a short period as a scientist and acquired multiple scholarships. She's an inductee of the Sigma Chi, a prestigious national scientific research society designed to honor those who have made noteworthy contributions in research. In addition, she has been bestowed the prestigious J.P. Leinfelder Award from Iowa Eye Scientific Meeting of University of Iowa. With that, I'd like to welcome Dr. Sharma to Zoom. Please feel free to share your presentation and begin. Thank you. Okay, you're muted, and we do see your presentation, and it's full screen.
Sorry about that. Thank you for the really kind introduction, Jimmy. I really appreciate it. So today I'm going to be talking about my research, which is out-of-body SANS research. And we are going to be using our ex vivo human translaminar autonomous system to study space-associated neuroocular syndrome pathogenesis. So something about this new model, which we have patented under patent application, is that it allows us to not only study different pathogenic paradigms that are associated with translaminar pressure on Earth, but now we can use this model for space-associated neuroocular syndrome as well. Okay. Alrighty. Okay, there we go. So what happens to our bodies in space? Basically in space, we are dealt with a lot of different detrimental factors like cosmic radiation, conditions of microgravity, stress, which includes emotional, physical, mental stress, which can come about due to insomnia, increased pressure, vascular pressure differentials, as well as tissue atrophy that we see. And so during missions or long duration space flights, we see a constant readaptation of not only different tissue systems like our brain, where there is neuroplasticity that is occurring, tissue atrophy in cardiac tissues, we become taller in space, as well as muscle loss, vascular changes, bone deterioration, which is why astronauts have to have an extensive exercise regime. But one of the things that does affect astronauts is vision loss. And that is which is a red risk factor for NASA, as well as TRISH, and that is why we pursued this proposal or project to study SANS. So space-flight-associated neuroocular syndrome, basically when we are under long duration space flights and when there's conditions of either no gravity or microgravity, there is cephalid fluid shift changes that occurs towards the brain. And gravity, which is what pulls the fluids downwards, is not there to counteract these forces in space. So what we see is an increased intracranial cerebral spinal fluid, and that causes increased orbital cerebral spinal fluid or intracranial pressure around the optic nerve sheath. This can cause a lot of detrimental factors, which include hyperopic shifts and optic disc edema, as well as globe deformations, flattening, and protrusion of the globe. And this can lead to either choroidal folds, cotton wool spots, optic disc edema, as I mentioned. So when there is normal pressure within the optic nerve or the intracranial pressure, fluid flows through around the sheath, your globe is perfect. But any increases, whether it is in the intraocular pressure or the intraocular region of the eye, or in the intracranial pressure region or the ICP region, that can cause damages to the optic nerve head. And when there is high or mildly elevated ICP, this can cause globe deformations, optic disc edema, and we see some of these other vascular structure changes, including choroidal folds and swelling of the optic disc, and optic nerve distension as well. So some of the things that can cause, the globe flattening can cause, is the movement of the plane of focus and defocusing of varied across the retina, as well as other visual stimuli, which is deprivation, light intensity, radiation, can all lead to changes which results in hyperopic defocus, retinal signaling changes, altered gene expression, scleral remodeling, and change in axial length. And how do we study some of these changes? We can study it not only at the retinal ganglion cell level or at the optic nerve level, but also at the optic nerve head, where we can see structural thickness changes, biomechanical properties or elastic properties of the optic nerve head changes, as well as the composition due to the extracellular matrix redeposition. So one of the risks or the SANS is considered an unexplained major risk factor. And so we would like to study the pathogenesis associated with SANS, not only at the optic nerve head and the optic nerve, but also at the retinal ganglion cell level, and how the retinal neurons are getting affected. So the gaps that are part of the SANS research is, how do we determine relationship between the fluid shifts, induced ocular changes, and the fluid shifts in the central nervous system, including whether elevated intracranial pressure or brain edema can it play a role. And to address these gaps, we need to understand what are the etiological mechanisms and contributing risk factors for the ocular structural and functional changes seen not only within flight, but post-flight as well, because these changes that are occurring under conditions of microgravity or lack of gravity can affect not only within flight, but post-flight for long-term effects. So is there a ground-based analog, as Jimmy mentioned, or are there models that can simulate the SANS syndrome? So currently the potential mechanisms of SANS include increased intracranial pressure. There could be translaminar pressure gradient, which is the change within the intraocular pressure compartment and the intracranial pressure compartment. And so that pressure differential that occurs at the optic nerve head is known as the translaminar pressure gradient. So is there changes within this translaminar pressure gradient that could be affecting the globe? Are there cephalothrombotic fluid shifts with volume increases that is affecting altered lymphatic drainage systems, the intracerebral volume, as well as cerebral edema, orbital and cerebral arterial and venous drainage defects, including one carbon pathway metabolism alterations that have been studied, choroidal volume expansion, hypercapnia related volume and pressure disturbances. But where do we come in, or where our project came in, was what is the exact etiology and pathophysiology? It is not known. So can there be changes within the intracranial and intraocular pressure gradients within the eye that could be affecting SANS? So our first path, or NASA's path to risk reduction, would include how do we preclinically study human tissue to evaluate pathogenesis and therapeutics ex vivo? And how do we identify the variability that is seen in SANS? Because not all astronauts have the same degree of symptoms. There is extensive variability not only in long-term, short-term as well as long-term space flight, but there is variability between astronauts. So can we look at environmental conditions? Can we look at genetics? Can we look at other detrimental factors or risk factors that could be associated with SANS, including comorbidities in these astronauts? Because in short duration space flights, almost 30% of astronauts see some sort of visual defect. But when you look at long-term space flights, almost 50% of astronauts are seeing some sort of visual defect, including one aspect, whether it's edema or choroidal folds or convulse spots or globe flattening or axial length changes. So the other aspect, of course, which is the long-term aspect, is that as we don't have a huge patient cohort between the astronauts, can we actually perform precision medicine in terms of countermeasures? If you're giving supplements to these astronauts, can we have every astronaut device therapeutics? And can we study that between one astronaut to another? And is there differences between the left eye versus the right eye? Is there differences between male and female astronauts? And how do we perform precision medicine in accordance to that? So our basic overview is, how do we take individuals, or astronauts in this case, perform disease pathogenesis or modeling of the disease using donor eyes or ex vivo human donor eyes? Can we use a model system to study this? And in the long run, can we perform high throughput testing for gene therapy, neuroregeneration, or just therapeutics in terms of supplements? The secondary aspect to this whole study was not only understanding the disease pathogenesis, but can we actually take skin fibroblasts from individuals, reprogram them to the stem cells and eventually the retinal neurons, transplant them into the donor eyes, and then go downstream and look at precision medicine as well. So before we started, and this was before we got involved with the TRISH project, I, as you know, I'm a glaucoma researcher. So we wanted to just look at human donor eyes, ex vivo, take their posterior cups, see if you could see patient cells into these posterior cups, and if we could assess translaminar pressure gradient. And that brought us to designing this unique, novel, innovative model system that can help us test drug therapies, compounds, and transplantation strategies in a preclinical manner, cost effectively and non-invasively as well. So this model system basically has two chamber systems. And as you are looking at the model, there is a dome shape where you can put a posterior eye cup onto this dome, and the optic nerve can go into this simulation of this ICP top chamber, basically. So this model system over here, and you can see the transparent view of the model. What it allows us to do is actually autonomously regulate not only the ICP chamber, but the IOP chamber as well. So both systems are independent of each other. And this is a unique model system because it allows us to keep these human donor eyes viable for 30 days in a sterile environment where we can not only study internal and external pressure testings, but we can infuse media into this model system to test therapies, drug targets, as well as biomarkers. So the opportunity of the TAS model is when we are looking at target selections or lead discovery molecules, it has to go through a pipeline to get into clinical studies. And this increases costliness of the drug therapies as well, because when you are testing therapeutics, you have to not only do in vitro studies, but as well as in vivo animal studies. But sometimes drug therapies that are tested in the in vivo animal systems do not or cannot relate to the human system. So how can we come in as an ex vivo human study system to test retinal therapeutics? And that is where we would like to insert ourselves into the clinical pipeline. So validating the model was our first key step. So we not only 3D printed the model, but we wanted to ensure that both the chambers, which is the ICP chamber and the IOP chamber, was independent of each other. It could be sealed. We could keep this human eye alive, pressurized, and in a sterile environment. And this is just a short video showing the procedure. So here you see a human eye, which is put into the system. The optic nerve, as you see, is right there. So we are using syringes to infuse media into the system. So there's an infusion port and an outfusion port. Then we put in the lid on top of that human eye where the optic nerve goes into that ICP chamber. Once that's sealed, then we go ahead and pressurize these syringes and we fill in the chamber. And this is just showing you the model in close-up. So there is the infusion ports, reservoirs, and the outfusion ports on the right-hand side. And that's the optic nerve that's within that chamber, ICP chamber. And the eye lies on that dome right there. So once we were able to completely seal that model, we could actually perfuse it, incubate it, and then using miniaturized pumps, we can actually, based on the flow rate, change the pressure with the pressure differentials within each chamber to a millimeter mercury of pressure differential. And the whole process from the start to the finish, from the dissection. So once we receive these human eyes, from dissection to all the way to modulation of pressure, only takes us 5 hours. So it is not a very hard system to set up. The next part was to study and see if the architecture of the human eye was maintained at 14 and 30 days of culture. So here I'm showing you cross sections of H&E of the optic nerve head at 14 days. And the architecture is pretty much maintained. You can see the optic nerve head and the axon and the fiber systems and the extracellular matrix is pretty much maintained within the human eye. And the blue staining represents the DAPI cells as well as the collagen 4, which is representing the extracellular matrix. This is true even at 30 days after culture. And then we wanted to see if we could have an average maintenance of pressure, which is the normal maintenance of pressure over a period of 30 days, which is within the range of translaminar pressure gradient of 2 to 5 millimeters of mercury. So we were successfully able to maintain the translaminar pressure of IOP with intraocular IOP average of 16 millimeters of mercury and ICP at 12 millimeters of mercury over a period of a range of 30 days. The next part was, can we modulate this translaminar pressure gradient? So what I had shown you previously was at normal translaminar pressure gradients, but what if we increase or elevate it in the intraocular region? So what you see is over time, from control to 7 days, there is extensive remodeling at the optic nerve head. When we elevate intraocular pressure, there's increased deposition or increased extracellular matrix deposition as depicted by collagen 4, where there's thickening of the beams. In addition, when you extremely elevate the translaminar pressure gradient by decreasing the intracranial pressure and elevating the intraocular pressure, what we actually noticed was there was extensive cupping of this optic nerve head as well. In addition, we don't RBPMS staining, which stains for the retinal ganglion cells. We see a decrease in retinal ganglion cells as well as restructuring of the collagen 4. In addition, over a period of 3 to 7 days, we see extensive gliosis at the optic nerve head when you elevate translaminar pressure gradient as well. And the lamellar deposition also increases over time. So this model system became the first of its kind. It could allow us to regulate both chambers independently. And that is how we got into the ex vivo paradigm to study SANS. And we got involved with the TRISH funded disruption, basically. So how did our connection to TRISH came about? So this would become the first ground-based analog to ex vivo mimic mildly elevated ICP in human eyes. Because one of the biggest things under conditions of microgravity is on Earth, when you're supine, seated, or standing, your ICP changes over the course of the day. And that allows, or the optic nerve head biomechanics is structured in such a way that it can allow that ICP changes throughout the course of the day. But when you go into space or during long duration space flights, when there is constant ICP that is being forced on the eye or the optic nerve head, that can significantly change the optic nerve head biomechanic. So what we wanted to see is TAS a suitable ground-based analog system to study the SANS phenomenon, and can we use this model system to define some of the parameters and the feasibility to understand SANS pathogenesis. TRISH got interested into our project because it was a high-risk project. It had never been studied for SANS. We were validating a novel preclinical system, and it helped us also to develop our prototype into a more structured, rigid system with less variability. Because initially, when we had started this model, we were changing our pressures by hydrostatic pressures by height, but the TRISH funding helped us not only streamline our prototype, but also incorporate pressurized model systems with small miniaturized pressure pumps. So there were two goals of the TAS model for SANS. One was to characterize the pathological changes occurring within the human posterior eye cup due to not only elevated IOP and ICP, but we wanted to add another dimension, which is, can we kink the optic nerve? Because due to globe flattening and the high pressure, the optic nerve, basically there is the tortuosity angle to the optic nerve, which could be affecting the retinal ganglion cells and eventually the hyperopic shifts or the changes in visual field defects that we see. The other aspect was, okay, is this retinal tissue alive to be able to transport, whether it is therapeutics or other molecules, and can we see the functional capacity of the retina within the human posterior eye cup after we can mimic the elevated IOP and ICP or optic nerve tortuosity after 14 days in culture within the TAS model system. So we did some variations of our model system for TRISH, where we basically put in inserts, and that allowed us to kink the optic nerve at 6-degree and 10-degree tilts. And this is just showing you the diagrammatical view of the ICP chamber, so that could allow us to tilt that optic nerve. So we wanted to test different experimental paradigms. So the first paradigm, which was our control, which was an average of the ICP and the IOP over a 24-hour period. So we looked at 12 millimeters of mercury as well as IOP at 16 millimeters of mercury. Group 1 became our mildly elevated ICP. So the ICP is not pathogenically elevated. That is what we see in idiopathic intracranial hypertension patients on Earth. It is just a mild elevation. But is that constant mild elevation pathogenic enough to cause SANS? So that is why we had that first condition where we had mild elevation at 15 millimeters of mercury, but a normal IOP at 16. The other thing is where we kept the ICP normal, but we elevated IOP to high levels, because when you exit Earth, there is elevation of IOP initially, and so we kept it at 21 millimeters of mercury and then brought it back to 14 millimeters of mercury with the translaminar pressure gradient of 2 to 9 millimeters. Group 3 became where we saw extremely high ICP, which is what you would see over 20 millimeters of mercury in IIH patients, but keeping the IOP constant with a translaminar pressure gradient of 6 millimeters. Then we had the mild elevated ICP, which is what you would see in conditions of microgravity with a 6-degree tilt and a 10-degree tilt. So major goals for the SANS validation was, can we keep these pressure maintenance for all these 6 groups, including the control, over a period of 14 days? Can we look at the globes pre and post culture and look at height, width of these globe changes, as well as could we identify retinal markers or gliotic markers, as well as inflammatory and apoptotic markers? Could we take the condition media out and look at secreted ECM proteins? Can we observe degeneration of axons, reorganization of the optic nerve head and the gliosis, as well as apoptosis in terms of tunnel staining, looking at functional transport of molecules, which is using CTB, as well as electroretinogram analysis for retinal functional as well. So the first aspect was maintenance of translaminar pressure gradients. So within all the 6 groups, we were able to have a constant pressure differential over a period of 10 to 14 days, with IOPs being measured every 24 hours for every hour within the 24 hours. And so we were successfully able to maintain these pressure differentials within all the 6 groups. The next part was we put the eye in, we took photographic images with constant distance of these posterior cups pre and post culture of 14 days. And this is just representative images of these posterior globes. Then our software engineer on board, Gaurav Sharma, he basically designed a software which was Tasmetrics that allowed us to not only look at the width of these posterior globes, as well as the height of these posterior globes over time at 14 days pre and post culture. So obviously, the tissue shrinks a little in culture, so this is just showing you the differences in shrinkage of these tissue over time between both the groups pre and post culture. In addition, we wanted to see the depth, because we were trying to mimic aspects of the globe flattening. Is there a depth increase in these ocular globes? So pre and post culture decrease in the post globes for groups 2, 3, and 5. There's a slight decrease even in the controls. And one of the limitations initially when we had started, just to give an aspect to that, is we had initially not been able to do this circadian rhythms of ICP IOP that we see sitting and standing over time as we see on Earth. So there was a constant pressure differential for the control groups. So this control was still a little bit towards the pathogenic side, because it was a constant ICP IOP when we had started this project, and that's where this is coming in. So then we normalized all our other groups, which is group 1s to 5, based on our control. And what we see is, there's definitely increased cell death occurring for retinal markers between groups 1, 2, and 3. And just to keep everybody in mind, the mild elevated ICP, the extensive increase in translaminar pressure gradient as in groups 2 and 3, as well as the kinking of the nerves in groups 4 and 5. So there's more survivability, of course, with mild elevation of ICP, but there's, of course, more apoptosis as well as gliosis in groups 4 and 5 due to the kinking of the optic nerve as well, which is what we can see with the apoptotic and inflammatory markers here. Next, this was not initially something that we had in mind because we were using hydrostatic pressures, but once we were able to use miniaturized pumps for each of the ICP and IOP chambers, what we were able to do is actually have a constant flow rate and keep the tissue much healthier and alive through the system. And because of this, we could actually collect conditioned media at the end. And that allowed us to look at the fibronectin expression or the extracellular matrix expression within the IOP and ICP chambers. And just to give a brief background on that, we are inserting the same media through the IOP and ICP chambers, infusion-wise. But even with that, we see a huge difference between the IOP and ICP chambers and the expression of fibronectin between both the chambers for each of these groups as well, with the IOP on the intraocular side having more fibronectin expression within the media or secretion of the fibronectin. In addition, even the collagen, there was an increase within the IOP chamber media or secretion of fibronectin in terms of the ICP chamber. And all of these have been normalized to the controls. What was also interesting to note is there was increased collagen deposition or increased collagen expression due to the optic nerve tortuosity in groups 4 and 5 as well. The next part was we wanted to look at optic nerve degeneration in the cross sections of the optic nerve post-culture. So what we did was we took the globes out of culture 14 days within all these 6 groups, and then we took cross sections 3 millimeters post the optic nerve head, in the pre-laminar and laminar and post-laminar regions. And this is just a depiction of that, and what the dark stained spots, which are identified by the arrowheads, represent the myelin staining by PPD or paraphenyl diamine stain, which stains myelin. So when you have healthy axons, you see a nice little ring around, or you see these little holes with nice little dark stainly myelin sheet rings. But once the axon degenerates or collapse, what you see is this pooling of the myelin, which stains really dark. Which is observed by these arrowheads in this diagram. So normalized to control, what we see is there is definitely an increase in degeneration due to the 10-degree tilt in optic nerve or kink compared to the other groups as well. And increased degeneration of these optic nerve axons over time. The next aspect was we took cross sections of these optic nerve heads after post-culture, and this is the H&E represents the morphological analysis, and due to the 6-degree and 10-degree tilt, we do see there's a curvature, the kinking of this optic nerve that's occurring. In addition, laminar deposition increases over time in the other groups as well compared to control. What we also notice is that there is increased gliosis in groups 3, 4, and 5, specifically due to the optic nerve tortuosity as well. There is slight increase in TLR4 expression amongst these groups, which is also seen in the control group as well. What next we wanted to see is take these cross sections of these optic nerve heads, and this is really zoomed in images, and look at the tunnel staining, which is tunnel stains for the DNA interaction after the cell starts dying. And so these green spots represent the death of or apoptosis of these cells over time. So what we do see as increase in group 1 as well as group 4 and 5. The other aspect was, can there be effective transport? So we infused chlorotoxin B, which is a molecule that can be anterogradally transported from the intraocular side. So we infused this media with CTB, which was stained for the red fluorescent marker or conjugated with the RFP or Elexaflor of 594. And what we see is at the optic nerve region, we do see increased red staining in the control, but groups 3 and 4 and 5 also showed essentially effective transport at the end of the optic nerve from the intraocular to the ICP region as well. So the biggest question was, of course, can we keep these retinas alive over a period of 14 days using these human donor eyes? So we were fortunate enough through OcuSciences, we're able to take pieces of retina post-culture, dark adapt them, and then put this in an ex vivo ERG system, which is something designed for human ex vivo retinal functional tests. And that is all collected to the electrodes, and we could measure at different amplitudes and intensities the retinal function. So this is a depiction of the control after 14 days in culture at different intensity. So the retina is alive after 14 days in culture in the control conditions. Next, we wanted to see if all our control group 1, 2, 3, and 4, and 5 were actually having retinal function. So as you can see, controls group 1 and 3 had A-wave responses, which is the mixed cone rod photoreceptor responses. But group 2, 4, and 5 gave us more of a flicker kind of response, and these are scotopic threshold responses in these retinas 14 days post-culture. So kind of conclusions from our part 1 was we were able to effectively maintain pressure over a period of 14 days to mimic some of these SANS conditions. We saw an increase in width with significant high changes, elevated inflammatory and apoptotic markers for optic nerve tortuosity groups. We saw increased extracellular matrix proteins among all groups compared to the control. There was a level of degeneration in the optic nerve axon, especially in group 5. There was the cupping morphological reorganization and gliosis that is observed in the experimental groups. In addition, there was increased tunnel staining in the mildly elevated ICP groups. Also, there was anterograde transport of CTB, which was observed in control high translaminar pressure and optic nerve tortuosity groups. And a functional ERG response was observed in control groups 1 and 3. So this led us to think, okay, what would be the future therapeutic and preclinical applications to reduce the SANS risk? Can we look at environmental conditions in the future, like elevated carbon dioxide concentrations, high salt content? Could we possibly test existing therapies, biomarkers, or identify new targets using some of these conditioned medias that we are seeing, or changes in the extracellular matrix that we are seeing from the ICP and IOP media that we are collecting? But before we could do any of that, or we can do any of that, what we wanted to actually test is, can there be effective cell transplantation, and can we do precision medicine so that we can address some of these things that we're talking about? So we wanted to see if we could utilize human retinal ganglion cells seated in these posterior cups, and can we keep these retinal ganglion cells alive within the TAS model over a period of time. So for this project, what we did was we took skin fibroblasts from control individuals, generated their stem cells, and Dr. Tawakkal on Tuesday actually gave a great talk on the stem cells and retinal organoids. But just a quick brief overview, we can actually induce pluripotent stem cells using Yamanaka factors, which is SOX2, CMYC, OCT4, and KLF4, basically oxys. So, and generate their retinal organoids, and that retinal organoids over a period of time from 25 to 30 days. When you dissociate them, actually that's the most opportune time for the retinal ganglion cells. And so by directed differentiation using conditioned media, we can generate these iPSC-derived retinal ganglion cells. This whole procedure approximately takes 3 months, and then we can seed these retinal ganglion cells on this posterior globes and put them in the translaminar autonomous system, pressurize it, seal it, and perfuse and incubate and modulate pressure. So before we start, of course, there's a huge cynicism involved with, can we actually culture adult retinal ganglion cells from our native retina from a human donor posterior cup? So to address that dilemma, we actually got human donor eyes from the Lions Eye Institute in Tampa, Florida, and we took retinal explants, dissociated them, and grew them in culture. And this is 7 days post-culture, where, you know, these all staining for RBPMS, as well as we saw neurofilament light chain expression as well, 7 days in culture from these retinal ganglion cells as well. So to actually truly proof of concept show that our iPSC-derived retinal ganglion cells can actually seed into these posterior cups, what we thought would be the best approach would be to take these human donor eye cups that we are getting from the lion's eye, can we decellularize them and remove all the cellular factors, but maintain the extracellular matrix composition or architecture, and then go ahead and seed our retinal ganglion cell, iPSC-derived retinal ganglion cells. So our first aspect was to decellularize these tissues. So just showing you, from one individual, the ODOS, and how we effectively did not see any RBPMS positive or retinal ganglion cells or GFAP after decellularization. Then once we did that, we kept them, in culture media, and while we did that, we had generated the iPSCs, derived the retinal organoids, dissociated that retinal organoids at day 30, and grown them in culture to generate these retinal ganglion cells, which are positively stained here for RBPMS and neuronal marker 2H1. And so you see this a nice neurite growth of these retinal ganglion cells. In addition, we also tested BRN3A positive markers, as well as looked at some of the gene expression of other retinal ganglion cells, as well as neuronal markers of these retinal ganglion cells. So the next aspect was to take these iPSC-derived retinal ganglion cells and seed them in the posterior cups. And once we did that, we put these posterior cups, kept them alive for a few days, and then once it integrated within the posterior cup, we put them in the TAS model for 7 days. And these are just cross sections, which I'm going to talk about in a few minutes. And we left them in 7 days within the TAS model once these posterior cups were seeded with iPSC-derived retinal ganglion cells. And this is just a video showing how we took those stem cells, infused them with Matrigel to give them like extracellular matrix to basically bind and integrate within the posterior cup. And once we did that, we left it in culture for 7 days, took this posterior cup and put them in the TAS model and maintained normal pressure gradient over a period of 7 days. This is cross sections of the decellularized cup where you don't see any laminin or GFAP expression. In addition, no collagen 4, the positive laminin and collagen 4 expression, so the extracellular matrix or architecture is maintained, but negative for GFAP and BRN3A. Once we recellularize them, BRN3A is positive for the retinal ganglion cells, which is what, is depicted here, but we did not see any, obviously no glial cells because we had decellularized these. So we only reseeded them with retinal ganglion cells, and that's what we see in these culture cups. So next we put them in the TAS model, and this is just a more zoomed in image of at the optic nerve head. What we see is the BRN3A positive cells, but there's no other markers. So the blue represents the cells in culture. So for the part 2, we actually used a combination of technologies, not just to prove that we can actually derive retinal ganglion cells, which is well researched now, but can we use these iPSC-derived retinal ganglion cells in these human donor eyes? As well as can we reseed them and put these human donor posterior cups with the successful culture in the translaminar autonomous system. So our next step in terms of precision medicine or based applications for the model would be to take these AV2 based GFP iPSC derived retinal ganglion cells. So basically, for our actual experiment, we would not, of course, decellularize these donor eyes because we would like to maintain the retinal structure and function, but instead take, whether we are trying to take it from astronauts or other individuals or patients, tag them with GFP, and then put them on these donor eyes and look at successful integration and regenerative capacity by putting in trophic factors, therapies, or other treatments. Can we look at translaminar pressure gradients in terms of optic nerve head biomechanics? But also the other aspect is because we're getting these donor eyes, which are human donor eyes, there's a huge variability in the optic nerve head. So we can also look at the heterogeneity of the optic nerve head using our model system. And of course, we would provide a preclinical humanized model to do this. Having said all this, there is, of course, limitations and to our model, but in addition to the limitations, and by overcoming these limitations, what would be the future vision of our model? So I did mention the variability of human donor tissue, and we as a human population, every one of us is so different, and there is such genetic heterogeneity within the population, which is what we see with our human donors that we get. But that also is beneficial to us because right now when we are looking at rodent studies, which are inbred mouse strains, we cannot identify sometimes that genetic variability, but this model system would allow us to look at that. We are using an exotomized human posterior eye cup, and that exotomy causes death of tissue, but because we have a control group built into our system, we can have normalization or basal level of apoptosis normalized to all our other experimental conditions. The long-term viability of our donor tissue was huge because initially when we had started, I had mentioned that we were using hydrostatic pressure pumps, but our mechanical engineer, Hussein Lohawala, was able to design miniaturized pumps that allowed a constant flow through the system. And this allowed us to not only keep the flow going, but it prevented us from doing the push and pull. So we had to go in and push and pull our media through every 48 hours. So our TRISH funding helped us immensely for actually increasing the opportunity of our prototype and development of our prototype as well. One of the biggest things, which, SANS is also, there is the aspects of vascular perfusion pressure. So we do not have that in our system, but it also works to our benefit because there has always been that aspect that is ICP or mildly elevated ICP, is that enough to cause SANS? And, there's huge reports that were saying that maybe ICP will not be relevant. But because our model only can delineate ICP and there's a lack of vascular, we can actually see that just mild elevation of ICP is that enough to cause some of the pathogenic effects that we see in our human donor eyes. And when we had started, we could not mimic some of the cyclic variations that we see in ICP and IOP on Earth. And so our control did show some slight elevations of the gliosis and other things that we see because there was a constant ICP and IOP. But now with our ability to modulate pressure, we can now insert a different control into our grouping where we can look at the cyclic variations and have that as a true mimic of a control. In addition, TRISH helped us increase the scalability of our TAS model. So our mechanical engineer designed these miniaturized pumps that are controlled, and our software programmer helped us design these Adreno boards that can actually regulate each chamber individually for each of these pumps. So now at a time, at one step, within one incubator, we can actually have 6 8 eyes going with 16 miniature eyes prompts, to each one for ICP-IOP for each of these model systems. Future directions would not only look at the therapeutic potential and the preclinical applications in terms of disease modeling and pathogenesis, but drug discovery through high throughput drug screenings, biomarkers, precision medicine, looking at medical history, genetics, lifestyle, environmental factors, as well as cell transplantation within our model system and genetic manipulations, whether it's in terms of CRISPR-Cas9 mediated genome editing or other gene therapies, AAV2 based gene therapies that we could look at within our model system. So the translational nature would look at ground-based experiments that could be done, but also because our model is so small, could we put it in conditions of zero and microgravity and change designs or effects on our SANS model? Would it be effective enough to be transported? So this needs to be studied as well. Can we test countermeasures, whether it's in terms of supplements, anti-gravity chambers, or other countermeasures? Would it be able to do human basis of testing? Because there is a huge unmet clinical need for SANS to identify some of these things for astronauts. So space health applications would include donor stem cells from astronauts, looking at the genetics of these astronauts, looking at the different pathogenic conditions causing SANS. We can compare right versus left eye posterior cups as well, male versus female. We can look at, ISS has elevated levels of carbon dioxide concentration. So can we infuse media that has elevated carbon dioxide? High salt diet is something because of the dehydrated foods. The concentration media could be infused with high salt concentrations. We can put it under zero gravity chambers in short and long-term flight studies, and basically collect condition media for biomarker expression to target future studies, and have a ex vivo human preclinical model system to test these therapies and then take them to the animal models and go downstream from there. So what is our future in terms of possible mission to moon and beyond? What we are envisioning with our engineering team as well as the other people in my lab is, can we actually downsize our system where we don't have tubing and pressure reservoirs? Instead, they are almost like IV infusion pouches that could be clipped on and off. So we could have the human donor eye set up on Earth, put it into a space flight mission, it gets taken in, and then through specific pressure modulating devices that are put on these system, can we have infusion of the media, and then the astronaut in space could actually clip on, clip off the ICP-IOP medias, and then we can test these when they come back to Earth to test different effects that conditions of microgravity can have on this human eye, and what other therapeutics or treatment strategies that we could have. So finally, I would like to acknowledge none of this work would have been without some of the huge help and benefits that we have and resources that we have, including Indiana University School of Medicine, especially Dr. Mao, who helped me with some of these optic nerve degeneration, Michael Peng, analysis of optic nerves, as well as our Eugene and Marilyn Glick Eye Institute, all the faculty, scholars, and staff for the help and resources that we have. Dr. Tucker, who provided the stem cells for us, Dr. McDowell, who helped with the innovation of this project, the engineers, Hussein Lohawal and Gaurav Sharma, who helped with some of our engineering aspects, as well as UNT Health Science Center, which helped immensely, Stacy, with some of or most of these experimental paradigms. She was instrumental for everything that we have done, as well as our histology cohort, as well as some of the other PIs and director of UNT Health Science Center. As well as none of this would have been possible without human donor eyes, which Dr. Amar's group has provided us, and we have a huge collaboration with their group, plus our funding resources, TRISH, which has helped us immensely to not only develop our prototype, but actually take it further into modulating pressure, circadian rhythms, all of these new avenues of resources. Thank you, and I am ready to take any questions that the group has.
Thank you very much, Dr. Sharma. That was an excellent presentation. And we have a few questions to ask, to address about certain topics that were brought up. One is about the sort of presumed etiology of SANS. And the question specifically is, what are your thoughts on the recent reports that insulin resistance is a root cause in SANS among astronauts?
Yeah, so I think that is something that we can definitely study. And there is, as I mentioned previously, there is a lot of pathological events that could be causing SANS, obviously. And so if we did have insulin resistance, can we actually look at, IGF receptors on these retinal ganglion cells? Can we look at all other types of factors that could be affecting the retina, as well as, obviously we don't have vascular perfusion, but we can just look at the retinal health and look at, glycolysis associated pathways, as well as diabetes associated pathways in general, and perfuse media in, which could help us study some of these factors or effects.
And actually along that line, bringing up the media, one of the questions related to the culturing conditions, and that is, how do you maintain the donor eye during those 5 days in TAM in regards to the temperature, the solutions, the media, the nutrients, and so forth? Can you comment on the culturing conditions?
Yeah, so it takes us only 5 hours. So as soon as we get the eyes from the Eye Institute, immediately that morning, we are always on call. So we immediately dissect the eye and put it in. And we have actually given all our neurotrophic factor infused media to the lion's eye already. So we ship out media regimens to them. So as soon as they get the eye on their end, they put it in the media, and these are normally corneal transplant eyes or posterior cups. So they're the most healthiest eyes that we get, or donor eyes that we get. So as soon as the cornea is taken, they basically put them in a sealed pressurized chamber that Lion's Eye has patented, and that has all the media and the nutrients, and they ship them out overnight to us. So we get them as soon as early in the morning, so within 24 hours to 36 hours post-mortem. And then once we get them, we keep them in sort of a bathing solution and put them on the model system. So if I get the eyes around 10 o'clock, and 5 hours is just for maintenance of pressure, but we can get it into the system and infusion of the media within 1 or 2 hours after we receive these eyes. So by noon, if I mostly receive them at 10 o'clock in the morning, by noon, we already have them up in the system and running. And they are kept at 37 degrees in an incubator. So all of these model systems, they're put into the incubator as soon as we put them in the chamber. And then there's tubing that comes out and connects to the pumps, basically out of the incubator.
This is an interesting question regarding future directions and countermeasures for eyes. Can any future directions include physical exercises for the eye and/or mental stimulus in terms of light, color patterns, which also exercise the eye and the optic disc?
So that is an interesting modality that we could pursue in the future. In vivo, obviously, there is, postural changes that happen in the astronauts because they are free floating or under conditions of microgravity. But as the orbit is fixed in the orbital socket, it would be harder for us to I'm trying to think if we needed to move basically the optic nerve, what would be the scientific benefit of that? But having said that, we could definitely look at light, because this model is transparent. So we could look at radiation, we could look at light effects. We could, in addition, look at aspects where we could put it in a device which would generate centrifugal force and have kind of a mimic, which is why we're trying to minimize all the tubing and the pressure reservoirs so that it's contained in just one unit system. So like have small little IV-like pouches that is within the system that just clips on, clips off. So we're not having these long tubings, we're not having these pressure reservoirs. So we could just have a small device system, and that can go into a centrifugal system kind of force device to mimic some of those conditions.
Could you talk a little bit about, or if you're aware of, how, say, cerebral spinal fluid flow and pressure are measured in low gravity environments? Are there challenges to that you're aware of?
Yeah, so right now, of course, there's lumbar punctures that are done, which are quite invasive. There are other different things like tympanic membranes now, can be used to look at ICP measurements as well as ultrasounds. You can do B scans. So look at optic nerve sheet diameter. Based on that you can kind of measure ICP. And so there is non-invasive ways to measure like Doppler and other ways, but they are within a millimeter of differential comparatively to, doing an actual lumbar puncture on these patients, which is the most invasive.
Going back to a little bit about biomechanical testing, is there any follow-on to do biaxial or circumferential biomechanical testing to obtain stress strain curves and see how sustained pressure increases will change the tissue constituents and therefore the tissue properties or behaviors?
Yes, so that is why we designed that software, which is the TAS basically. So what we were trying to do, so I did have, I measured the radius. So basically, we have prototype 3D printed a specific model that where we sit the eye in, and that is at a specific height. So we take, using a smartphone, we basically take the eye's picture and the exact way it was set before and after culture. And what that allows us to do is not only measure the radius, but also the length from the point of cut where we we cut the eye at the equatorial region. And so from that to the optic nerve head, what is the length of that eye? So kind of not obviously from the cornea, the axial length would not be accurate, but from the point of the cut to the optic nerve head, if each eye, individual eye, is having a difference, right? Pre and post culture.
And again, sort of going back to the etiology and the ability to assess that with the technology, could you, or are you planning to, or have thought about assessing the hypothesis that SANS is associated with swelling of the microvilli and the subarachnoid space surrounding the optic nerve, and this is induced by reduced eNOS production?
Yeah, so some of those, obviously eNOS and, DOSMAD and a lot of other things have been, for normal tension glaucoma and other things have been used. And we have not, because, this model got just developed last year, or we filed a patent application last year. So that is our future. That is what we would like to do. We would like to look at other pathogenic effects. We would like to look at eNOS effects. We would like to pursue all those intracranial pressure related or vascular pressure perfusion related drugs and therapies to look at disease pathogenesis.
And one question regarding the cells that you do use in your culture systems. Since the eye has so many different types of cells, can you just comment briefly on the cell types that are utilized in your culture systems again?
Yeah, so that is why we decellularize the model, because, obviously the retina is made up of a lot of different cell types. So what we wanted to just see is if the stem cells that we were deriving were retinal ganglion cells. So we looked at the retinal function of these retinal ganglion cells, as well as that is why we didn't want the effects of glia and all that affecting, but we wanted to decellularize the model to look and seed iPSC-derived retinal ganglion cells to see successful integration. But having said that, in the future, we will not be doing that. This was just a proof of concept thing that we did just to see if the cells that we had generated, could they be integrated. And they are iPSC-derived retinal ganglion cells, which is, the retinal neurons in the innermost layer of the retina, in the retinal nerve fiber layer, and not the other retinal cells, to see if they could integrate within the human donor eye. But for future therapeutics or future clinical transplantation studies or preclinical transplantation studies, we will just tag the cells that we derive and culture with either different fluorescent markers like GFP. So when we integrate it into the donor eye cup, we can identify these retinal ganglion cells that are fluorescing green are actually coming from our culture system versus the donor eye retinal ganglion cells. So that's how we would basically distinguish the two different cohorts.
Well, we look forward to seeing how this technology and the advances as you move forward, as well as your findings towards mitigating this risk. And we'd like to thank you very much for your time and your presentation today. It was an excellent presentation, and we're very much appreciative.
Thank you so much. It was a pleasure being here, and I look forward to working with TRISH, hopefully in the future, or NASA, and looking at some of these things, because this has really piqued my interest. I'm a glaucoma scientist, so for sure, going into space health definitely changes a different disease paradigm that I can study.
And I think that really highlights the need to involve people with the clinical backgrounds and different backgrounds than just studying and applying those experiences to mitigating risk to human health in space. So thank you so much for your presentation and your efforts.
Yeah, thank you so much. Have a good day.
All right, with that, thank you everyone for attending. And please join us tomorrow at 12 p.m. Eastern, where we're going to be hearing from Frederick Zenhausen, who will be talking about the new test strip.
Thank you.
Thank you.
Have a good day, and thank you to TRISH.